So first we'll talk about hormone independent versus hormone dependent characteristics. So within hormone independent characteristics, these are known as your primary sexual characteristics, which include your X and Y chromosomes, your gonads, which are your sexual organs, ovaries in the females, testes in the males, hormone dependent characteristics, which are your secondary sexual characteristics. And your hormone independent characteristics produce the hormone dependent characteristics, which are different in both males and females. So they include your genitalia, your brain, the hypothalamus, pituitary gland, other structures, and hormones as well. And then certain behavior for other organisms such as courting, mating, nest building, nursing, rearing, etc. So hormone independent characteristics and give rise to your hormone dependent characteristics. So now we're going to talk about asexual versus sexual reproduction. So obviously, as most people know, the main limitation of asexual reproduction is that it limits genetic diversity. However, the main benefit of asexual reproduction is you don't need another partner and it's much quicker than typical sexual reproduction. So there's different types of asexual reproduction. There's budding, which is a growth through meiotic cell division, and then it buds off the parent, so it's like a small little like bud coming off the parent through an outgrowth. For example, is Hydra. There's also regeneration, where damaged tissues are regenerated, even to the degree of growing a new limb. For example, some reptiles, some lizards, etc. If they're missing a tail, they're able to regenerate that tail. And some starfish, if you cut off a piece of a starfish's arm, it's able to regenerate into a whole new starfish. Fission as well, for example, sea anemones, and then pathogenesis, which we'll talk about as well, which is a production of an organism from an unfertilized egg through sexual behavior. So it's still asexual reproduction, but it occurs through sexual behavior. So we'll talk about that. An example is a whiptail lizard. So as mentioned before, for budding, like you see here, this is an outgrowth coming from the original parent. So a new individual forms from this outgrowth. So this is just the beginning stage, but a new individual will form from this bud right here. And then regeneration, um, the this right here, this leg piece was cut off from a sea star, and it was able to regenerate completely brand new legs and regenerate into an entire animal. So some animals have both asexual and sexual reproduction cycles. It depends purely on their environment. So for example, aphids have both a sexual reproduction cycle, which occurs in the winter, which is as typical. They under males, females undergo sexual reproduction, produce eggs that gives rise to their offspring. However, they also have an asexual reproduction during the summer cycle. During the summer cycle, they probably have lots of resources. During the summer cycle, they probably have lots of resources. So because they have lots of resources, they want to make the most of those resources. So they rapidly reproduce asexually to produce as many offspring as possible. So over the winter, they'll undergo sexual reproduction, but during the summer, when environmental conditions are favorable, they'll undergo asexual reproduction in order to reproduce as quickly as possible. And so this is what I was talking about with parthenogenesis. So some animals can act as both male and female during reproduction. So here, whiptail lizards that undergo parthenogenesis are all female, but they take turns acting as either the male or the female during reproductive behavior. And so this is necessary in order for ovulation to occur. And by acting as a male and a female, it ensures that parthenogenesis can occur here. So as you see here, um, when the whiptail lizard acts as a male, obviously their estrogen levels decrease, but when they act as a female, their estrogen levels increase, which increases the chances for ovulation to occur. So that's why some female lizards they act as the female so they can undergo ovulation, while another female lizard acts as a male to ensure that the egg will get to ensure that the egg will be released, and then they take turns. So it will be the opposite as well. The female lizard acting as a male will eventually act as a female, while the female lizard acting as a female will eventually act as a male in order to ensure that eggs are released for parthenogenesis. So now we're going to talk about sexual reproduction in animals. So there's three fundamental steps that occur. Gametogenesis, which is making the gametes. So that is your sperm and eggs. Spawning or mating, where the gametes are brought in together. And then fertilization, which is the actual fusion of the gametes into zygotes. So we'll start with the easy question. Which statement about asexual reproduction is true? And like I mentioned previously, you could just chat it to me directly.
Some people say still taking the class. I totally blown this
I
stuff.
think
Yeah,
someone's
maybe it's
unmuted. All right. Getting a few answers in. All right. So the answer is C. Obviously, it allows single individuals to produce offspring. Certain cases, like in parthenogenesis, you need a partner, while in other cases, like in aphids, for example, with oh, why is mine? Sorry, I have to go back. In aphids, for example, they don't need a partner. Like here in the summer cycle, they can just undergo asexual reproduction and reproduce that way. So, yep, the correct answer is C. It allows single individuals to produce offspring. The bottom answer, I saw some people put E. Offspring are always diploid. That's not true. Not all organisms have diploid offspring. Some organisms have haploid offspring, which eventually mature into diploid offspring. So now we're going to talk about the first step of sexual reproduction, which is gametogenesis, specifically spermatogenesis for males and oogenesis in females. So for spermatogenesis and oogenesis have a similar structure. So keep in mind the 2N and N. Once again, 2N means diploid, N means haploid. And we go from 2N to N during meiosis. So first, there's a mitotic part for spermatogenesis. You have a male germ cell. Germ cells can become any cell. So that male germ cell undergoes mitosis and becomes a spermatogonium. At that point, spermatogonium, they're self-regenerating, so they can keep undergoing mitosis amongst itself to keep producing more spermatogonium. But eventually, that spermatogonium division gives rise to a primary spermatocyte. So we're still in the diploid stage. But then that primary spermatocyte undergoes a meiotic division. Now we're starting to go through meiosis. So the primary spermatocyte goes through meiosis 1. Remember, meiosis has two parts. So primary spermatocyte undergoes meiosis 1, and it turns into a secondary spermatocyte. Now it's haploid. Eventually, that secondary spermatocyte goes through meiosis 2. Secondary spermatocyte becomes a spermatid, and that spermatid eventually matures in the testes into sperm. So definitely memorize the names of each step, because a question on the exam could be, what is the order of spermatogenesis? So the correct answer would always be spermatogonium, primary, secondary, spermatid, sperm. But then they'll try to switch it up. But spermatid could be first, or spermatogonium could be last, etc. So definitely memorize the order for spermatogenesis. And as I mentioned previously, oogenesis is similar. You also have a diploid stage and a haploid stage. You have, the way you can think about it, you have technically three diploid stages, where once again, female germ cell undergoes mitosis to become a oogonium. That undergoes mitosis to become a primary oocyte, and then that undergoes meiosis. So the primary oocyte and the primary spermatocyte both have primary in the name. Those are both diploid, and then the primary undergoes meiosis to become a secondary. So primary oocyte undergoes meiosis 1 to become a secondary oocyte, and in a secondary oocyte, it also has a polar body. Then that secondary oocyte once again undergoes meiosis to become meiosis 2 to become a ootid, and that ootid eventually matures into an egg. And that's how you get your gametes. So now we're going to talk about the process of fertilization. So in fertilization, there are several little rules and events that need to be followed. So first, the sperm and the egg have to chemically recognize each other. You can't take the sperm of one organism and the egg of another organism and combine them. Well, typically, you can't combine them. So they have to chemically recognize each other. The sperm has to be activated, which allows it to gain access to the cell membrane of the egg. The cell membrane of the egg fuses with the cell membrane of a sperm. The egg blocks entry of additional sperm. You don't want multiple sperm fusing with one egg. So as soon as one sperm fuses with one egg, that's it. The egg does blocks entry of any additional sperm being able to fuse because that's more genetic material being brought in, which obviously the zygote would not be able to eventually undergo mitosis. The egg is metabolically activated and stimulated in order to start development. And lastly, the egg and sperm nuclei fuse to create a diploid nucleus of the zygote. So there's lots of regulation of fertilization. There's species specificity, where sperms fertilize eggs of the same or highly closely related species only. So human sperm will only be able to fertilize human eggs. Obviously, there are special instances where, for example, they've taken the sperm of a lion, egg of a tiger, etc., and then managed to fuse them together. However, even those offspring are still not able to reproduce amongst themselves. So that's another way fertilization is also regulated in that if you are able to produce an offspring using sperm and eggs of unrelated species or of not the same species, their offspring will not be able to reproduce. So species specificity is important. Cell specificity. So the sperm is only going to fertilize an egg. A sperm is not going to fertilize the cell wall of a random cell in your body. It'll only recognize an egg and fertilize that. So not other cells they encounter. And then a number specificity in that, as mentioned previously, only one sperm fertilizes an egg. You can't have multiple sperm fertilize an egg. So where does production of sperm occur? So the production of sperm occurs in the male reproductive organs, specifically in the testes. So I'm not going to go over the male reproductive organ or the female reproductive organ, what is it? The structures themselves, but definitely memorize these structures. There could be a, what is it? A labeling section on the exam will they'll have an image of the male or female reproductive organ and they'll tell you, hey, um, label this part, what's this part, etc. So definitely memorize where everything is located. But I will talk about where specifically spermatogenesis takes place. So spermatogenesis takes place in the seminiferous tubules of the testes, and that is where sperm is produced. And then you also have what are known as Leydig cells. So Leydig cells are found adjacent to the seminiferous tubules and the testicles, and then Leydig cells produce testosterone. And then that testosterone is produced when you have high amounts of luteinizing hormone or LH. Luteinizing hormone is part of the endocrine system, and the endocrine system is really important in here as well. So, as mentioned previously, spermatogenesis takes place in the seminiferous tubules. You have what are known, so your male germ cells undergo mitosis to become spermatogonium. They undergo mitosis again to become a primary spermatocyte here. And you have what are known as Sertoli cells. So the Sertoli cells basically cover and protect these sperm cells as spermatogenesis takes place. And another important part of the Sertoli cells, they envelop, nourish, and protect the developing sperm cells. And so, once again, primary spermatocyte undergoes meiosis to become a secondary spermatocyte. That secondary spermatocyte undergoes meiosis to become a spermatid, and they mature into sperm. And as sperm mature, they move from the outer layer of the seminiferous tubule towards the center, and then they're eventually shed into the lumen right here. So, if you guys were here for chapter 38 and 39, on Monday, you guys may recognize this diagram on the right. Remember what I mentioned, how the endocrine system, everything, pretty much every hormone that's part of the endocrine system follows the same pathway. So as you can see here, the hypothalamus, oh my, give me 1 second. The hypothalamus produces gonadotropin releasing hormone. Then the gonadotropin releasing hormone activates the anterior pituitary. The anterior pituitary gland produces luteinizing hormone and follicle stimulating hormone. These then stimulate Leydig cells, which produce testosterone, which is important for your reproductive tract and other organs. Follicle stimulating hormone stimulates Sertoli cells, which provides the nourishment and protection for the developing sperm cells. And these Sertoli cells obviously help with spermatogenesis. And it could also produce inhibin. Inhibin, as is found in the name, it inhibits the anterior pituitary to make sure that follicle stimulating hormone doesn't produce anymore. So the negative feedback loop as here or over here as well. If you have lots of testosterone, testosterone can act as a negative feedback loop to the hypothalamus or the anterior pituitary in order to make sure that more testosterone is not produced. So as it says here, testosterone feedback inhibition of luteinizing hormone is stronger before puberty, weaker after puberty. Luteinizing hormone stimulates your Leydig cells to produce testosterone. Follicle stimulating hormone and testosterone stimulate your Sertoli cells, which provide the nutrients and production for spermatogenesis to be sustained. Inhibin provides negative feedback to inhibit FSH release, and then testosterone inhibits gonadotropin releasing hormone release. So for oogenesis, once again, this is the female reproductive tract. Um, Try to memorize the structures of the female reproductive tract. So the ovarian cycle, once again, you have your primary oocytes. Your diploid primary oocytes undergo meiosis one to become your secondary oocytes, and then that secondary oocytes become a ootid, and then eventually matures into an egg. And so during ovulation, the secondary oocyte is in prophase of meiosis two, and it remains there. And then if fertilization and implantation has occurred, what you have here is known as the corpus luteum. The corpus luteum is maintained if fertilization occurs. However, if fertilization does not occur, the corpus luteum starts to degenerate. Once the corpus luteum degenerates, this starts the cycle of your primary oocytes being formed again into secondary oocytes, ootids, eggs, etc. So if fertilization occurs, corpus luteum is maintained. If the corpus luteum is not maintained, then the cycle starts again. Or if fertilization does not occur, corpus luteum degenerates, and then the cycle occurs once again. So now we're going to talk about the follicle itself. So follicular cells proliferate in response to FSH stimulation, and then they provide nutritional support for the growth of the oocyte, and they also secrete estrogen. So after ovulation, these follicular cells become the corpus luteum, and the corpus luteum secretes progesterone. And progesterone and estrogen are both really important for fertilization and also the growth of a baby if fertilization occurs. In that when there's not enough progesterone, a miscarriage can happen. And so even for uh your ovaries, there's also same thing with the endocrine cycle. Hypothalamus stimulates the anterior pituitary with the gonadotropin releasing hormone that then produces luteinizing hormone, FSH, which stimulates the ovary. Ovaries produces estrogen and progesterone, and that then stimulates your uterus. And then everything can act as a positive feedback loop where it stimulates the anterior pituitary or stimulates the hypothalamus to produce more, or it could act as a negative feedback loop as well. So negative feedback regulation occurs throughout most of the ovarian cycle, and then your luteinizing hormone and follicle stimulating hormone levels remain relatively constant except during ovulation. And then in the middle of your ovarian cycle, and then around a few days before ovulation, luteinizing hormone surge occurs. When that luteinizing hormone surge occurs, you also have positive estrogen feedback, which enhances the release of uh, the gonadotropin releasing hormone, more LH, more FSH. And then if you have the failure of a positive feedback loop and luteinizing hormone surge, ovulation does not occur and egg is not released. So this is the hormonal changes that occur during the ovarian cycle. I definitely say memorize these graphs. When I took the class, I had a question that basically had one of the hormones like cut out, and the question was, what is the hormone that's missing from the graph? So, as mentioned previously, during ovulation, your luteinizing hormone surge occurs. So that's how you're able to recognize this is your luteinizing hormone. Around 14 days of your uterine cycle, that's when ovulation occurs, and then that's when your luteinizing hormone spikes. Follicle stimulating hormone remains relatively consistent. And then here, so negative feedback inhibition of gonadotropin releasing hormone by estrogen keeps your FSH and LH levels low right here between days 0 and like 10. So it keeps it low. However, eventually you have positive feedback excitation. Eventually, instead of inhibition, there's positive feedback. And then that positive feedback excitation of gonadotropin releasing hormone produces a huge luteinizing hormone surge, and then obviously a smaller follicle stimulating, oh, a smaller follicle stimulating hormone surge in the midst of the ovarian cycle, so that stimulates ovulation. And eventually, as mentioned previously, if fertilization does not occur, your corpus luteum starts to uh, degenerate. So if you want to test for ovulation, if you want to test if you're ovulating correctly, the good way to test is to see what your levels of luteinizing hormone are. If your luteinizing hormone levels are extremely high, that means ovulation will probably occur. If your luteinizing hormone levels are low, you will not have ovulation. So, let's say fertilization occurs. Now we're going to talk about pregnancy. And then, obviously, if fertilization occurs, your endometrium lining doesn't shed, corpus luteum does not degenerate, the corpus luteum is maintained. So over the course of pregnancy, your human chorionic gonadotropin level is maintained, but your estrogen and progesterone levels increase steadily. And the estrogen to progesterone ratio also increases slowly, where you're going to have more estrogen than progesterone. And then there's estrogen and progesterone, as mentioned previously, they play extremely important roles where if you don't have as much progesterone, a miscarriage can happen. And they both play extremely important roles for um the zygote, and that so the estrogen is produced by the placenta. It stimulates endometrial and mammary duct growth, and it also suppresses prolactin and lactation, while progesterone is also produced by the placenta, but also by the corpus luteum. That's why you don't want your corpus luteum to disintegrate if fertilization occurs. So your progesterone, it stimulates endometrial growth, it suppresses luteinizing hormone and follicle stimulating hormone release. The reason it suppresses that is because you don't, if one of your egg gets fertilized, you don't want more eggs to be released. And it also suppresses myometrial contraction. So we talked about this a lot. In women, a drug that blocks the luteinizing hormone surge would most immediately prevent blank. We have one answer so far, two. Mhm. So most of you got the correct answer. The answer is ovulation. So as mentioned several times, if you're ovulating, that's characterized by an increase in your luteinizing hormone surge. So I saw some people put a peak in estrogen. The reason it's not a peak in estrogen is because as you see here during ovulation estrogen goes down. Here estrogen does go up, but you have to remember um here, estrogen does go up, but during ovulation itself, estrogen does go down. Luteinizing hormones remains relatively consistent throughout. So if luteinizing hormone is blocked, that most directly prevents ovulation from occur. And as you see here, Luteinizing hormone doesn't stimulate estrogen. Estrogen stimulates luteinizing hormone and FSH release. So the correct answer would be B, ovulation. So now we're going to talk about controlling fertility. So there's several methods of contraception in order to control fertility. And then over here you see a failure rate. So numbers of pregnancies per 100 women per year. You could think of it as a percentage. If you have unprotected sex, it has an 85% failure rate, meaning an 85% pregnancy rate. There's also different methods here, which all still have high failure rates. Obviously we have condoms, jellies, cervical caps, which still have a decently high failure rates as well, but obviously much better than unprotected. And lastly, there are also hormones. Hormones provide the best methods of contraception, and that oral hormones have a 0 to 3% failure rate, non-orally administered hormones less than 1%, and then progestin only pill taken within 72 hours after unprotected sex has a 5 to 40% failure rate. Obviously, it's taken within 72 hours, so if you take it almost immediately, it'd be closer to 5. The longer you wait to take Plan B, it would take obviously it would your failure rate chance increases. And then there are also implantation blockers, IUDs, mifepristone, which is a drug. These also have extremely low failure rates. And then vasectomy, tubal ligations, ways of sterilization. Obviously, those would be the lowest failure rates. So now we're going to move on to chapter 42, which is animal development. So we're going to talk about the basic animal development. After a zygote is formed, what happens? So you have what's known as cleavage. So cleavage is that early cell division occurs, but no cell growth occurs. So the zygote just starts to divide amongst itself. So the embryo becomes a solid ball of small cells, and then you have what's known as a blastocele. So it eventually cleaves and cleaves, and then a blastocele is a central fluid-filled cavity that forms within the ball. So right here is your blastocele, and then the embryo goes from a zygote, cleaves into this 8-cell stage, and then cleaves even more to become a blastula. And then within the blastula, each individual cell is called the blastomere, and the middle fluid-filled cavity is called a blastocele. So this pattern of cleavage varies within with different species. So with eggs with little yolk, you have complete cleavage, such as in frogs right here. The cleavage is almost like every blastomere is similar in size because of complete cleavage. So in frogs, the vegetal pole has more yolk, while and then division is unequal, and daughter cells in the animal pole are smaller. So animal poles, blastomeres are smaller right here, while the vegetal pole, which would be here, the blastomeres are larger. And then you also have incomplete cleavage, such as, for example, in zebrafish. Incomplete cleavage occurs in eggs where you don't have a lot of, oh, they do have a lot of yolk, but because they have a lot of yolk, when the zygote starts to cleave, the cleavage furrow cannot go through the egg completely. Like you see here, the cleavage furrow is able to go through the egg completely, or not the egg, the zygote completely. But here it's not able to go through it completely because it has a lot of yolk. So as a result of that, there's incomplete cleavage, and then there's also discoidal cleavage. In discoidal cleavage, the embryo forms as a blastodisc right here, and it sits on top of the yolk at the bottom. Then you have superficial cleavage. So this is typically in insects. So it's a variation of incomplete cleavage where your plasma membrane starts to grow inwards around your nuclei and form cells. So like you see here in fruit fly, you have mitosis. So the nuclear division starts to occur, but without cellular division. So the cell itself is not dividing, but you have lots of nuclei being formed. So eventually that results in a s- s- uh, syncytium, which is a single cell, also has many nuclei. So here you started with one nuclei. The nuclei itself starts to divide. The nuclei itself divides. You have lots of nuclei, but the cell itself does not divide. So here you have a syncytium, so single cell with many nuclei, and eventually these nuclei migrate to the edge of the cell membrane. So they're all located in the middle. They start going through the edge, and then cellularization occurs, creating a blastoderm. So in mammals, we're gonna talk about fertilization specifically in mammals now. So in mammals, fertilization occurs in the oviduct. So cleavage occurs as the zygote travels from the oviduct to the uterus. So the sperm travels all the way from here into the oviduct. That's where fertilization occurs. And then eventually the zygote will travel down to here. So within the uterus is will they'll actually start to implant, then they start to develop. So here, site of fertilization, they start moving down and then they're implanted in the uterus. And that's when cleavage starts to occur and the zygote starts to develop. So implantation occurs when the trophoblast adheres to the endometrium. So the endometrium is the uterine lining. So implantation of the blastocyst. So implantation that occurs in the oviduct is an ectopic or tubal pregnancy, and then the zona pellucida normally prevents this. So now what happens to the blastula after cleavage occurs? So the blastula eventually turns into a gastrula through gastrulation. So gastrulation is the movement of cells which transform your blastula into an embryo that has multiple tissue layers and multiple body axes. And each of these tissue layers all have a function in terms of what eventual part of your body they're going to produce. So in triploblastic animals, three germ layers form. The ectoderm, endoderm, and argentera. So in mesoderm, which would be eventually here. So ectoderm, endoderm, mesoderm. And obviously, as you know, in triploblastic animals, three, so three germ layers are going to form. So as mentioned previously, each layer has its own specific thing it's going to form. So your endoderm layer becomes the digestive lining, respiratory tract, pancreas, thyroid, liver. So endoderm is the one that's all the way over here. Your ectoderm, which is your outer layer, that becomes your nervous system, eyes, ears, and your skin. While your mesoderm is the middle layer that contributes tissues to your heart, blood vessels, muscles, and bones. So what's on the outside produces typically what's on the outside of your body, your eyes, your skin. Mesoderm, the middle, that produces all your a lot of your internal organs, and an endoderm is all the way on what's the inside of your body and produces the lining of your digestive tracts. So it makes sense. The order of the ecto, meso, endoderm is the order of how your body works as well. So over here, this shows how that actually occurs. So the vegetal pole of the blastula starts to flatten out, and then some cells will start to change shape, move inwards to form the archenteron. Um, Once that archenteron starts to form, you have what are known as primary mesenchyme and then secondary mesenchyme. Cells start to break away, becoming primary mesenchyme, and then other cells start to break away, forming secondary mesenchyme. And then these secondary mesenchyme have extensions which attach to the overlying ectoderm. So right here, secondary um mesenchymes create these attachments that attach to the very top or your ectoderm. So that why does it attach to the top? It pulls in the archenteron. So it pulls in the archenteron, which elongates it, and then eventually the mouth will form where the archenteron meets the ectoderm, and then the blastopore will form the anus of the mature animal, and then these start to develop into what we have the ecto, endo, and mesoderm. So your archenteron cells extend, flatten, and migrate over one another to form a long, thin tube. And as mentioned previously, this is where your mouth forms and the blastopore becomes the anus. And so gastrulation produces an embryo with three germ layers that all influence one another through inductive tissue interactions. So then there is also organogenesis, which is the actual production of your organs and organ systems, and then neurulation, which is the initiation of the nervous system. And neurulation occurs very early on in organogenesis. So as you see here, you have your ectoderm, mesoderm, endoderm. So a group of cells passes over the dorsal lip and that becomes the chorda mesoderm, and it produces a rod of mesoderm called the notochord right here. So that produces the notochord. That notochord provides support for the embryo. And then the chorda mesoderm induces the ectoderm, your ectoderm right here. The chorda mesoderm induces the ectoderm to form your nervous system. So steps in neurulation, the ectoderm lying over the notochord. So the ectoderm lying over the notochord here eventually thickens and forms the neural plate. Once that neural plate is formed, the edges start to fold within each other and then a deep groove forms. So here a deep groove starts to form. These folds then fuse next to each other. So the ectoderm folds fuse. That produces the neural tube and the layer of ectoderm that produces the neural tube as you see here because your um notochord starts to combine and then eventually forms that neural tube, and then you have a top layer of ectoderm on top. And eventually these neural tubes and neural crest cells give way to your nervous system. So right here. Now you have a top layer, which is your ectoderm, and then the neural tube and the neural tube and neural crest cells. You can't see the neural crest cells here, but as you see here, these will eventually become the nervous system. So the neural crest cells dissociate from your neural tube, start to migrate outwards, and they lead the development of the connections between the brain and spinal cord with the rest of their body. So they start to produce your peripheral nervous system. And they also are responsible for producing lots of diverse structures, such as your jaws, your skull, your face, pigment cells, glands, smooth muscles, and as mentioned previously, your peripheral nerves. So homeotic genes control body segmentation, and they all have a DNA sequence known as the homeobox. And in vertebrates, Hox genes are homeotic genes, part of your homeobox, that control differentiation among the anterior posterior body axis. So here, uh, the genes closest to the 3' end are expressed in the anterior most positions. So in your homeobox, the 3' end will give rise to parts of your brain, such as your hindbrain, midbrain, forebrain. However, here, HOXB9 will give rise to your spinal cord, which continues all the way to your posterior end. So those closer to the 5' end will produce your posterior end. And so the homeobox helps segment your body to know that which gene is responsible for which part of your body. And as it says here, Hox genes are expressed along that anterior posterior axis in the same order as their arrangement on the chromosome. Different segments receive different combinations, which then eventually act as transcription factors. So hypoblast cells form the chorion, and then that chorion eventually creates the placenta, which develops, and from that chorion and uterine tissues, the placenta is extremely important because the placenta exchanges nutrients, gases, and wastes between the mother and embryo. So that's another reason why they say, please make sure you don't smoke or drink during pregnancy because anything that you eat or drink during that time, any nutrients, any gases, any waste will be exchanged with the embryo during that time. So the epiblast produces the amnion. The amnion surrounds the embryo and it's filled with amniotic fluid. And so once that amnion is ruptured and your amniotic fluid starts to be released, what's typically known as water breaking, that's what causes labor in humans and causes contractions and the baby to, or the embryo slash the baby to eventually come out. So allantoic tissues contribute to the formation of the umbilical cord right here. The umbilical cord is how the embryo is actually attached to the placenta. And then Blood vessels in the umbilical cord carry nutrients and oxygen from the mother to the developing fetus and also carries away wastes. So it provides nutrients and oxygen to the fetus and then releases carbon dioxide and urea from the fetus. So gestation or pregnancy is around 9 months, and you have trimesters. So in the first trimester, which is only around 4 weeks, the embryo becomes a fetus. The heart begins to beat by the fourth week, and by the eighth week, the limbs start to form. So during the first trimester, you have lots of rapid cell division, tissue differentiation, and that the first trimester is the most actually one of the most important parts of pregnancy because the first trimester, the fetus is extremely susceptible to radiation damage, chemical damage, pathogen damage, etc. because the amniotic fluid, um, the umbilical cord, the chorionic placenta, everything is still starting to be formed at this point. So as a result of that, the first few weeks or the first trimester is when you really need to be the most cautious. And so embryos can be damaged before the mother even realizes that they are pregnant at that time. So the second trimester, the limbs start to elongate, fingers, toes, facial features form, and your nervous system starts to develop. And then your third trimester is when your internal organs mature. And then once the lungs are officially mature, that's when birth occurs. And the baby is able to obviously survive without the need of the placenta, umbilical cord providing nutrients, oxygen, etc. So there are lots of serious effects that could occur from exposure to environmental factors through pregnancy. As mentioned previously, exposure to alcohol and cigarette smoke, or even protein malnutrition, they can result in low birth weights, mental disorders, developmental complications. And so that first trimester is really the most important in order to make sure that the fetus is able to develop properly. So we're going to move on to the last chapter now, which is chapter 43, the nervous system. So what are the major functions of the nervous system? So some of the major functions of the nervous system, interaction of the external and internal environment. So you have your sensory nervous system and your motor nervous system. Sensory is what you're able to detect from the environment. Motor nervous system is how you produce actions in order to respond to the environment. It regulates their responses to the external environment. You have autonomic control, which are your reflexes, and then voluntary control, which is what you actually think of while moving. And your nervous system is also important because it's integrated with other organ systems and organs. So it helps regulate these systems and it makes sure that homeostasis is maintained. So organization of the nervous system. As mentioned in chapter 42, the nervous system starts to form from the notochord and the neural crest, and that eventually becomes the central nervous system and peripheral nervous system, as we see here. But there's also nervous systems in other animals as well. So the sea anemone has a nerve net. So the nerve net is obviously not as sophisticated as human nervous system, but it provides simple behaviors such as contraction, relaxation. Earthworms also have a nervous system through their ganglia, but they have ganglia in each segment, so that helps coordinate movements. And then their anterior ganglia is like considered part of their brain, so that helps control more complex behavior. Squid nervous system was extremely important for us as it helped us discover action potentials. So in squid, more complex behaviors are served by a collection of neurons in specialized ganglia that process and integrate information. Their giant axon is really what helped us discover how action potentials work as humans and how the nervous system works. And so with humans, we have the central nervous system, which is our brain and spinal cord specifically. And then this communicates to every other cell and organ of the body via the peripheral nervous system. That's why any damage to your spinal cord essentially means that parts of your body will never receive any signals from your brain ever again. So protect your spine, protect your back, etc. Your central nervous system is crucial, and then that helps talk to your other organs through the peripheral nervous system. So, as mentioned previously, central nervous system is your brain and spinal cord. Peripheral nervous system involves ganglia, your nerves, and receptors that are throughout your body. And so you have sensory systems. Sensory systems are your receptors, your afferent nerves, which are approaching your brain, and then sensory nerves. You have your motor system, which is your output. You have higher and lower motor centers. You have your efferent nerves exiting the brain. So if people are always confused about afferent nerves are your nerves where you're able to detect input from your environment. So it's sending information to your brain, approaching your brain. Efferent nerves are nerves that are sending information to the rest of your body in order to tell your body what to do in response to the outside environment. So efferent nerves exiting your brain. Oh, someone had a good acronym in chat. Sensory afferent, motor efferent. That's a good acronym. Same, yeah. And then you also have your muscles and glands, which are part of your motor system. And then within your nervous system, you have your autonomic nervous system, which is, like we mentioned, automatic. You have your sympathetic nervous system, fight or flight. If you remember, we discussed this on Monday. And you have your parasympathetic nervous system, which is rest and digest. And then you have higher association and integrative centers. So the nervous system is comprised of two types of cells. You have your neurons, which are your nerve cells. These nerve cells help generate and conduct the electrical signals or the action potentials. And then you have glia as well. Within glia, you have macro and microglia. Macroglia modulate neuron activity. They provide support to your neurons. For example, oligodendrocytes, Schwann cells, or astrocytes, which are your immune responses in your nervous system. And then you also have microglia. Microglia are also similar in that they're small phagocytic cells derived from blood stem cells, and they help maintain your synapses. The synapse is the area between two neurons that is where neurotransmitters are released in order to bind to the second neuron. So your synapse is extremely important and it needs to be maintained by your microglia. So structure of a neuron. So you have dendrites. Dendrites are what actually receive information from other cells. So this is the presynaptic cell and postsynaptic cell. Oh, you have two neurons here. So the presynaptic cell is sending in information to the postsynaptic cell. Right here is your cell body or your soma, where your nucleus is. The axon is where that signal or the action potential is being sent out. And then the axon is being sent out. Right here is the axon terminal. Terminal, as you know, that's the end. That's the end of the axon. That is where your signal will be released. The synapse is the area in between your presynaptic cell and your postsynaptic cell. Within the synapse, these neurotransmitters and the signal is being released from the axon terminal so that that neurotransmitter can bind to the postsynaptic cell and the process starts all over again. And so the neuron's axon right here, the axon needs to be myelinated. And the reason it needs to be myelinated is that it helps provide obviously protection for the axon, but most importantly, it makes sure that your signal is being sent much faster. So the Schwann cells produce right here. Schwann cells produce myelin, which is a cell membrane that provides electrical insulation for your axon. So over here, you have intervals within your axon known as the nodes of Ranvier. And so the electrical signal is able to jump from one node to another node. So the myelinated portions, you have Schwann cells, they produce myelin, they help provide electrical insulation among the axon. Yes, I'll post the PowerPoint slides eventually. It'll be on the Queens College Learning Commons website by the end of today or tomorrow. Sorry, someone said that in chat. So as I was saying, you have Schwann cells which produce myelin, and then they insulate huge portions of your axon. The myelinated portions are known as your nodes of Ranvier, and within these nodes, your electrical impulse will be able to jump from one node to another. So your electrical impulse will go down much faster throughout your axon. Oh, uh-huh. So within your nerve cells, you have lots of morphological diversity. As you see here, you have uh Purkinje cells in your cerebellum, where you have tons and tons and tons of dendrites. Over here in the cerebral cortex, you also have dendrites. It's not as combined as the cerebellum, but in your cerebral cortex, it's much longer. And then you have your retina. In your retina for bipolar cells, your dendrites are extremely small, and the actual neuron itself is extremely small as well. So all these morphological differences just show that your mammalian nervous system has adapted based on the specific functional adaptations. So in your Purkinje cells, you need lots of dendrites. In your cerebellum, you need lots of dendrites in order to receive information and then send it out. Cerebral cortex, you need your dendrites, but you need it to be much longer. You need the neuron to have an extremely large axon to be able to transport information around or on a larger distance. While your retina, the information is right there. It's right in front of you. So for your retina, for bipolar cells, the neuron itself does not need to be as long. The dendrites itself does not need to be as long. So your body has adapted based on all of these your body has adapted your neurons, and they all have completely different morphological differences because of that. So now we're going to talk about electric potentials and action potentials. So we have what's known as patch clamping. So a patch clamping is where an electrode can be made from a glass pipette that helps conduct electrical charges, and one electrode is placed inside the neuron, another electrode is placed outside the neuron, where you can measure the differences in voltages over time. So you can measure how action potentials change it. And then over here, you could suction off you have a patch clamp which could suction off a part of the neuron, and after it suctions off a part of the neuron, you could detect the ion channel within that neuron. So if a single ion channel is maintained within that patch, you can measure the voltages within that ionic channel, because the ionic channel will maintain the current within that little clamp that it had. So you could tear that ionic channel away from the cell and then determine what the current is like. So like you see here, when it's closed and when it's open, your ionic current obviously changes. And so we're going to talk about ionic gradients across the membrane. You have an unequal distribution of ions across the cell membrane. Intracellular, you have a lot more potassium, while extracellular, you have a lot more sodium and chloride. So once again, a lot of potassium inside your cell, a lot of chloride and sodium outside the cell. And so this is a result of an electrochemical gradient. So typically what happens, this is a typical concentration gradient. Typically what happens, if you have a lot of potassium outside the cell, a little potassium inside the cell, etc., potassium will go where there's a little of it. That's the typical concentration gradient. If you have a lot on one side, a little on another side, as you see here, concentration diffusion force, your potassium will go towards the right in order to make it even, as you see here. If there's a if there's a similar amount on both sides, your concentration diffusion force will be equivalent on both sides. However, you have what's known as a charge repulsion force. And so this is what's known as the electrical chemical gradient, and that's what maintains the high level of potassium within your cell. So the charge repulsion force is actually stronger than your concentration gradient. As you see here, you have a high positive concentration outside the cell, and you have a negative concentration inside the cell. So despite the amount of potassium being equivalent among both sides, so despite your concentration gradient being maintained, because there's an unequal voltage gradient, this potassium will go towards the left, towards the negative. So the charge repulsion force will go towards where there is more negative in order to maintain the voltage gradient, even though the concentration gradient here is not maintained. Like you see here, the concentration gradient isn't maintained because you have a lot of potassium on the left. However, the voltage gradient actually takes precedence over the concentration gradient in order to make sure that your voltage is equivalent on both sides. And so negatively charged macromolecules have a high concentration of potassium on the inside of the cell. That's why, as we see here, you have 140 potassium intracellular millimolar, while you only have 5 extracellular. And that is due to this voltage gradient. So you have what's known as a sodium-potassium pump, membrane ion pumps. So ion pumps use ATP in order to transport ions against the concentration gradient. So the main function of your sodium potassium pump is to keep intracellular sodium low. So sodium will be taken outside of the cell, sodium will be taken from inside the cell to outside the cell, potassium will be brought inside the cell. And so this is against the concentration gradient because uh obviously you have a lot of potassium inside the cell, but you're still bringing more potassium inside. And that is due to the voltage gradient. Your sodium potassium pump acts as a result of that voltage gradient in order to bring in potassium against its concentration gradient. And obviously it uses ATP in order for that to happen. So you have 3 types of gated ion channels. You have ligand gated channels, voltage gated channels, and mechanically gated channels. Ligand gated channels are channels where a receptor has to bind to that channel in order to open it. Voltage gated channels are channels that are typically closed due to whatever voltage is maintained. However, if the voltage increases, then that channel is open. And then you have mechanically gated channels where some sort of pressure is put on that receptor channel in order to open it. So as you see here, during resting potential, your resting potential is typically around -60 to -70 millivolts. So during resting potential, only your leak potassium channels are open. What that means is that potassium channels can leak outside the cell. So your voltage gated sodium channels are closed, sodium can't be brought in. Chemically gated potassium channels are closed, potassium can be brought in. You have potassium channels, which allows potassium to go in and out. So your resting potential is -70. Eventually, a signal comes in. When that signal comes in, a signal comes into the neuron. That signal causes the beginning of the action potential, which causes the cell to depolarize. When a cell becomes depolarized, its membrane potential goes up. So what causes its membrane potential to go up? Over here, your sodium channels start to open. So a signal comes in. That signal causes your voltage gated sodium channels to open, and that causes sodium to rush into the cell. Because sodium starts to rush into the cell, now you have a depolarization where your neuron has an extremely high membrane potential, and it's going to send that action potential down the axon. Eventually, the voltage gated sodium channel will close. When that voltage gated sodium channel closes, sodium will not be able to be brought into the cell anymore. So your intercellular membrane potential will not go up anymore. But chemically gated potassium channels start to open. When chemically gated potassium channels start to open, potassium starts to go out the cell. Potassium starts to go in the cell and out the cell. So a lot more potassium starts to go out the cell, and your sodium potassium pump works as well. Your sodium potassium pump helps make sure that sodium goes outside the cell, potassium gets brought into the cell in order to re-obtain your initial membrane potential. And so hyperpolarization specifically is when your membrane potential goes below your resting potential. So chemically gated potassium channels start to open, potassium goes in, some potassium channels go out the leak channel, and then your sodium potassium pump helps make sure that the inside of the cell returns back to initial membrane potential. So once again, this is exactly what we talked about here with the opening and closing of action potentials. Right here is your resting potential. That's when voltage gated sodium channels are closed, voltage gated potassium channels are closed. The only thing that's open is your leak channels. Then a signal comes in. That signal causes one of your voltage gated sodium channels to open. Sodium starts to rush in. When sodium starts to rush in, that causes a depolarization inside the cell. Depolarization increases your intracellular membrane potential extremely high. You go from -60 to 50. More sodium channels start to rush in. As more sodium channels start to rush in, eventually depolarization, a hyperpolarization needs to occur. You don't want your membrane potential to be extremely high because that'll mean it's overstimulated. You don't want your signal to be sent continuously. So once that happens, your voltage gated potassium channels start to open. When voltage gated potassium channels start to open, potassium rushes out the cell. Potassium rushes out the cell in both the leak channel and your voltage gated potassium channel. As it rushes out the cell, um, your voltage gated sodium channels close as well, so that causes an undershoot. You go way below your resting membrane potential. But eventually, thanks to the sodium potassium pump, the sodium that's inside the cell will be taken out the cell, but the potassium that's outside the cell will be brought into the cell, and that will cause you to return back to resting membrane potential. So propagation of the action potential, this is just showing how they use the patch clamp method in order to detect how the actual potential travels over time. So they had a point A, point B. At point A, over here, you see the actual potential was here. As time went by, at point B, the actual potential came there. So here, sodium ions enter at point A. Because sodium ions enter at point A, that's the start of the action potential. Inside is getting positive. But in time 2, sodium ions flow down the axon towards point B. And at time 2, sodium ions enter point B. Point B actually depolarizes, and then that's the actual potential is generated at point B. So this is just showing how at point A and point B you have actual potentials. So here's what I was talking about with saltatory conduction in myelinated axons and the jumping effect within the nodes of Ranvier for action potentials. So once again, sodium channels open, generating an action potential. This sends a current from this node down to the second node of Ranvier. Once the current is sent down to the second node of Ranvier, the second node starts to prepare itself. So the second node starts to prepare itself by bringing it to threshold. So once that threshold is reached, an action potential is reached. And the threshold is important because action potentials are a action potentials are a 100% or nothing type of part of your body. So what that means is that action potentials, they either reach threshold. If threshold is reached, action potentials occur. If it does not reach threshold, action potentials will not occur. No matter how close uh your intracellular membrane potential is to threshold, if threshold is not released, you will not release the action potential. So you're not going to release 50% of an action potential, 70% of action potential, etc. It's 100% or nothing. So the current from point A or node A will be sent to the current at node B, bringing node B to threshold. Then the upstream sodium channels inactivate, causing a refractory period at node A. And at node B, the action potential is able to be jumped from node A to node B. And now node B sends the action potential, and this continues from here to here to here. So that's what causes the actual jumping. So now synaptic transmission, what actually occurs in the synapse, right here is the actual synapse. So synaptic transmissions translate electrical signals into chemical signals and then back into electrical signals. So what you see here is acetylcholine. Acetylcholine is a neurotransmitter. And so the signal goes from the axon into your axon terminal, and now you have lots of acetylcholine, which is your neurotransmitter. So sodium rushes into the cell. Once sodium rushes into the cell, that tells the acetylcholine, hey, we need to be sent out of the cell. We need to be sent out of the axon terminal into the postsynaptic cell. So acetylcholine will be put into a vesicle. After being put into a vesicle, it will bind to the membrane of the axon terminal, and then the vesicle will fuse with the membrane terminal and then send the acetylcholine out. When the acetylcholine is sent out, the acetylcholine will bind to receptors on the postsynaptic cell. So this is the presynaptic cell, this is the postsynaptic cell. Acetylcholine will bind to the membrane of the axon terminal, or the vesicle will bind to the membrane of the axon terminal, sending acetylcholine out. Acetylcholine will bind to receptors on the postsynaptic cell. Once it binds to receptors on the postsynaptic cell, as you see here, acetylcholine binds here, sodium gets brought in, potassium gets brought out. What happens when sodium gets brought in? An action potential occurs. So that's how action potentials are continued on. The neurotransmitter or the signal gets sent out into the synapse, and at the synapse or the synaptic cleft here, it will bind to receptors on your postsynaptic cell, and then that will cause action potentials once again. So you have what's known as spatial summation and temporal summation for your postsynaptic cell. Spatial summation occurs when you have several postsynaptic potentials arriving at the same exact time. So you have several different postsynaptic potentials arriving at the axon hillock here at the same exact time. So you have multiple different action potentials causing an increased. How do I go back? Oh, sorry about that. I think I accidentally ended slide. Okay, here. So spatial summation, you have several different action potentials all summing up together. Temporal summation, at the same synapse, you receive multiple postsynaptic potentials, but they're all the same postsynaptic potential. So at the same synapse, you receive multiple postsynaptic potentials. So all those postsynaptic potentials can be summed up into one. Essentially, spatial summation, uh you have several different potentials arriving at the axon hillock, while temporal summation is at the same synapse, you have multiple of the same potential. And so this is just showing how your nervous system as a whole works. So with the knee-jerk reflex, with the, as someone mentioned in chat, the same sensory afferent, motor efferent. So a hammer stretches the tendon in the knee, stretching your receptors in the extensor muscle. This then fires action potential. The red is your afferent, it's approaching your brain. This fires action potentials to your brain. So in a monosynaptic pathway, the sensory neuron synapses with the motor neuron reach the ventral horn of your spinal cord. Then the ventral horn will then release efferent potentials in the motor efferent pathway exiting the brain. So the motor neuron causes action potentials to the conducts action potentials to the extensor muscle that eventually causes contraction, and then a multiple synapse pathway inhibits firing in the motor neuron for the antagonistic muscle, which causes your leg to extend. So everything works in conjunction. This one simple knee tap causes your motor neurons to reach your brain, and then your brain is able to send action potentials back down to that same muscle to tell your body what to do, all without you thinking. This is completely this is completely natural or automatic. And that's everything. I'll stop sharing now. So yeah, that was chapter 41, 42, 43. Um, if you guys have any questions, want me to re-explain any of the slides, let me know. Otherwise, yeah, have a great rest of your day, everyone. Yeah, so the PowerPoint will be posted on the Learning Commons website eventually. You guys are really welcome. Please fill out the feedback form, and the recording will be available on the YouTube by the end of today or by tomorrow latest.